Hello friends, this video on molecular basis of inheritance part 27 is brought to you by examfear.com. No more fear from exam. So now we will try to relate genetic code with mutations. Now we have spoken about mutation in our previous lesson where we saw that if there is a change during say DNA replication or there is any change in the sequence of the nucleotides, it can lead to mutations, and these mutations over a long period of time can lead to evolution. So when we talk about mutations, we have discussed about two types of mutation. One is the point mutation and the other one is the frame shift mutation. So when we talk about point mutation, it is about a change in a single base which affects only a single codon. So that means in this case, all other codons remain unaffected. So only one base is being affected. So let us look at some examples of point mutations. So let us take this example. So here you see, this is the nucleotide sequence which we can see. So now if we want to know the amino acid sequence, so what will be the amino acid sequence? AUG is for methionine, UUU is for phenylalanine, UGC that is for cysteine, UAC is for tyrosine, UCU is for serine, CUU is for leucine, and GUC is for valine. So this would be the sequence of amino acids. Now let us suppose there is a change in a single base. Now if I say the change, what is the change? Let us suppose that in this UAC, here we have UAC, let us replace this A with G. So if we replace A with G, what will happen? So what would be the new amino acid sequence? The new sequence would be, see, all of these will remain unaffected. But this UAC earlier was tyrosine, but now it is UGC. And what is UGC? UGC is cysteine. So other than this, the entire sequence will remain the same. So you see tyrosine gets replaced with cysteine. But when one amino acid also changes, so the kind of protein which you will get out of this and the kind of protein which you will get out of this will differ. So this is how point mutations occur. Now it is not only replacing one particular say base. We can also look at this example. Let us take this example where you have this sequence. So if you write down the amino acid sequence, this is how it would be. Aspartic acid and then isoleucine and phenylalanine and phenylalanine. So this would be the actual sequence. Now here also if we bring about a change, let us suppose here instead of UAC, so here if you see this is UAC, now if we replace this C with U, so instead of C we put it as U, then what would happen? So here if you see the others will remain the same, but earlier UAC was for tyrosine and now UAU. Now here we see that UAU and UAC, both of them are for, they both code for tyrosine. So here you can see UAC and UAU, both of them code for tyrosine. So in this case earlier also it was tyrosine and now also it is tyrosine. So this type of mutation where even though there is a change in the base but the result is not affected. So this type of mutation is known as silent mutation. That is, silently the base has been changed but there is not much impact of that on the phenotype. So this is called silent mutation. Let us take yet another example. So here again let us suppose we have this sequence. So if we try to find out the amino acid sequence, this is how it would be. AGU is for serine, UAC is again for tyrosine, UUC is for phenylalanine, UUU phenylalanine, UUU again phenylalanine, and UUC is also phenylalanine. So this would be the amino acid sequence. Now here let us suppose the change which we bring about is here in this UAC, instead of C, this C is replaced with G. So now if we replace the C with G, what happens? So the initial part will remain the same. But A, what is UAG? So if you see here, UAG is a stop codon. So basically after this, there is a stop codon. Now since stop codon is encountered, so after this, they will not even be read. So all these codon will not even be read. So the entire process of translation will stop here. Right? So the deciphering of genetic code will stop here because we encountered a stop codon. So these are some of the examples where we saw that the change takes place in just one base of the codon, but that also have an impact on the amino acid. So these are examples of point mutation. So here we can study mutations in terms of the genetic code, and this brings about the changes in the phenotype. Now let us talk about the frame shift mutations. Now there are certain mutations where a particular change can not only bring about a change in that particular codon and that particular amino acid, rather the entire chain of amino acid gets changed. So here addition or deletion of a base which changes the reading frame, thus affecting the amino acids being synthesized. The entire reading frame gets changed. So we will look at it. So let us first talk about a scenario where addition of a base can cause frame shift mutation. So let us take the first example. So this is the sequence. So let us try to write down the amino acid sequence out of this. So this will be methionine. UUU would be phenylalanine, UGC will be cysteine, UAC will be tyrosine, UCU would be serine, CUU would be leucine, and GUC would be valine. So this would be the sequence of amino acid that we can decipher from this genetic code. So now let us suppose that the change which it encounters is if we insert an additional base on a position. Let us suppose we insert an additional base at this position. So let us suppose at this position we insert A, that is adenine is being inserted here. So what would be the new sequence? Now the entire sequence is going to be changed because we want a codon will always be a group of 3. So now after this U and U, you have A, so it will become UUA. So now everything gets shifted. So now the next one will be UUG, that is UUG. And then the next one would be CUA, then CUC, and so on. So this is how it would be. This is going to be CUA, then CUC, then UCU, then UGU, and so on. So now what is happening in this case? If you see, the first one is still remaining methionine, UUA. What is UUA? It is going to be leucine, and then again leucine, again CUA is again leucine, then finally serine, and then cysteine. So now if you look at the sequence of this amino acid and compare it with this, so both of them are completely different. And why so much of difference arised? Because here also you just inserted one base, but it changed the entire reading frame. Because of the insertion of that one base, the entire thing got shifted, and that is why it is called frame shift mutation. So in this case, you see a total change in the amino acids. Now let us take another example where you have the sequence. So if you try to write down the amino acid sequence, this would be something like this. This would be tryptophan, UAC will be tyrosine, then GAC is aspartic acid, then isoleucine, then phenylalanine and phenylalanine. I hope you are able to understand how am I writing this. Now here we will look at an example of deletion. So if we delete a base, so which base we want to delete? Let us suppose we delete this G. So we want to delete the G. So this G we have deleted. So once we delete the G, what would happen? The new sequence would be AUG, UGU, ACG, ACA, UUU, UUU, and so on. So this is how the new sequence would be. So this would be methionine, then again UGU would be cysteine, ACG would be threonine, ACA will again be threonine, UUU would be phenylalanine, UUU again phenylalanine. So again, if you compare it with this, there is no match at all, because in this case as well, the entire reading frame has been shifted. So this is also an example of frame shift mutation. However, if the number of bases which are added or deleted is a multiple of 3, then the impact is less. So let us look at an example of that sort. So for example, let us take a scenario where we add or delete the number of bases which is a multiple of 3. Now let us suppose in the same example, instead of deleting only G, let us suppose if we delete UGG. So we delete the entire UGG. So in that case, what will happen? This UGG will vanish, but if you look at the amino acid sequence, in that case, the sequence will remain the same, just that this will not be there. So one amino acid will not be there. So whenever the addition or deletion doesn't impact the entire reading frame, then we say that the impact is less, because at least the same amino acids are getting synthesized. But whenever the change is such that it is impacting the entire reading frame, so altogether a complete different sequence of amino acid will be synthesized. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.